Taking a look at the cell cycle. Before we look at the cell cycle, a few things we need to review first. Cells reproduce by dividing and passing their genes to their daughter cells. So you have an original cell that's going to go through the process of mitosis, resulting in 2 new daughter cells, and these daughter cells will be genetically identical to the original cell. The nucleus always divides before the cell divides. This is important because the nucleus is going to control the cell's activities due to it containing all of the genetic material. Cells must be able to grow and divide to keep organisms going. Nuclear division combined with cell division allows cells and whole organisms to reproduce themselves and to grow. All cells in your body are genetically identical, except for the gametes, the reproductive cells. All cells are derived from one cell, and that's the zygote. A zygote is the fusing of 2 cells, one from mom and one from dad, together. When a zygote starts to divide, that is mitosis, results in 2 identical cells, and then those 2 continue to go through mitosis, and the process continues until the full organism is developed. Before division in eukaryotic cells, thread-like structures called chromosomes become visible in the nucleus. The photograph is a set of chromosomes in a human male. Each chromosome is 2 chromatids held by a centromere. The centromere would be where they're connected, and it's in a different location on the different chromosomes. Note the different sizes of the chromosomes and the position of that centromere. The sister chromatids are each individual part. The number of chromosomes depends on species. For humans, there's 46 chromosomes. Flies have 8 chromosomes. When you look at the diagram of the chromosome, you have his telomere that's at the end. There's also telomeres on this end of the chromosome as well. Telomeres are protective sequences of nucleotides found at the ends of the chromosomes. They become shorter each time a cell divides, which results in an individual organism's aging process. Some cells are able to replenish their telomeres by using the enzyme telomerase. Cancer cells can do this and remain immortal. In other words, cancer cells can prevent themselves from dying off. Keeping the enzyme telomerase active can help keep the cells alive. There's a lot of new research going on with the telomeres, and we'll talk about them a little bit more in detail later in the notes. Chromosomes are able to be stained so that they can be studied. A karyogram is the study of a whole set of chromosomes arranged in pairs by size and position of their centromere. The photograph you're seeing here is representing a karyogram. A karyotype is just the number of chromosomes and what they look like, the size of the bands and the centromere placement. They haven't been arranged in a karyotype, where in a karyogram they are arranged in pairs. Taking a look at the structure of chromosomes, you have the chromatids. There's 2 of them. One of 2 identical parts of a chromosome held together by a centromere formed during interphase by the replication of a DNA strand. The centromere is going to allow the 2 chromatids to be held together. It could be anywhere along the length, but its position is characteristic for a particular chromosome. You also have your telomeres, which are at the ends. And then the genes for different characteristics. Each one of these is representing different genes. DNA is the molecule of inheritance and is made up of a series of genes. Each gene is one unit of inheritance. DNA molecule is about 2 nanometers wide, and the total length of the DNA in the 46 chromosomes of an adult human cell is about 1.8 meters. And it's all packed in a nucleus with a diameter of about 6 micrometers. There are different protein molecules that prevent the DNA from getting tangled into knots. The DNA is wrapped around these different proteins. The proteins are histones, and chemically they're basic, while DNA is acidic. Two other essential features for nuclear division are the centromeres and the telomeres. Again, looking at chromosomes, you can see on each of these chromosomes the different genes have been highlighted. All right, a few questions for you guys to answer. If you would please pause the video and take time to answer. Matching pairs of chromosomes are called homologous pairs. In humans, there are 23 pairs of chromosomes, one set from mom and one set from dad. So looking at your diagram, you have homologous pair of chromosomes at the top, and then after replication, there's homologous pairs of chromosomes that contain the sister chromatids. In looking at the karyogram, you'll notice that the first 22 pairs are numbered 1 through 22. You see the numbers. Those are the autosomes. The autosomes represent all of your chromosomes except for the sex chromosomes. The sex chromosomes are not numbered. They're labeled X or Y. In this example that we're looking at, we have an X and a Y. That represents a male. If we had two X's, then it would be a female. Then we have haploid and diploid cells. Cells that contain two sets of chromosomes are described as diploid, as represented by 2N. Think diploid, that die at the beginning represents 2. The N is the number of chromosomes in one set. A cell which contains only one set of chromosomes is described as haploid, which is represented just by the letter N. These would be the gametes, which makes sense. If gametes combine to make a zygote, they would need to be haploid so that when the two are fused together during fertilization, we would have a complete set of chromosomes. Looking at a chromosome after replication, you have the double structure made of two identical structures called chromatids. They are joined together at the centromere. During interphase, the period between nuclear divisions, each DNA molecule makes an identical copy of itself, the chromatids, which join again at that centromere. So this is one chromatid, this is a second one, and they're joined at the centromere. Each chromatin contains one molecule of DNA. DNA is the molecule of inheritance made up of a series of genes, which is one unit of inheritance. One gene's code for only one polypeptide involved in a specific aspect of the functioning of an organism. When you look at the genes found on the chromosome, if this gene represented eye color, then the duplicated pair, they would both represent eye color. If this was representing curly hair or straight hair, then both of these genes would be the same. The gene for a particular characteristic is always found at the same position or locus. So when you look at a pair of homologous chromosomes, you have your gene here at a specific position. When we have the chromosomes with their homologous pairs, this would be considered a pair of alleles. This would still represent the same gene. Again, if this was eye color, then this one would be eye color. It doesn't have to be blue eyes. It could be brown eyes and then blue eyes. The total number of different genes in humans is thought to be anywhere from 20,000 to 25,000. There's two forms of chromatin. Euchromatin, which stains lightly. The DNA histones complexes are loosely formed together, and it's used in protein synthesis. And then the heterochromatin cannot be transcribed, so it cannot be used in protein synthesis. It stains darkly, and is found on the peripheral part of the nucleus. Each member of the homologous chromosomes comes from one of the parents. There's 23 maternal chromosomes and 23 paternal to give us a complete 23 homologous pairs. Each member of a homologous pair possesses genes controlling the same characteristic. That pair of genes would be known as an allele. Remember, they're at the same locus, so you could have a set of genes or a set of alleles that's representing eye color. One of them could be blue, but the other one could be representing brown eyes, but they would be in the same locus or same position on the chromosome. Cystic fibrosis is a condition caused by a faulty allele or a mutation of a gene that codes for a chloride channel protein needed to produce normal mucus. A faulty allele causes a very thick mucus. Both the homologous chromosomes have to show that faulty or mutated allele for the individual to have cystic fibrosis. If only one of the chromosomes has the mutation, then the individual would not have cystic fibrosis. That individual would be considered a carrier. Looking at nuclear division, there's a lot of growth that has to happen as a zygote matures into an adult. With about 1 trillion cells, each cell is genetically identical. This type of division is called mitosis. So when you have an egg and a sperm that unite together, that's fertilization, and then that forms the zygote. The zygote then begins that growth and dividing through a process known as mitosis. Once a zygote forms, the zygote has to go through mitosis in order for the zygote to become an embryo. Well, sex cells have to go through a second division before fertilization can occur, and that's known as meiosis. Meiosis is a second type of cell division, which is going to take the number of chromosomes and half them to make them haploid cells. Looking at the cell cycle, it's a regular sequence of events that takes place between one cell division and the next. There's 3 distinct phases: interphase, the nuclear cell division, and then cell division itself. Interphase is the first part of the cell cycle. It's where the cell spends most of its time. There's 3 parts to interphase. G1, the G stands for gap. The cell grows to normal size after cell division. It makes RNA and enzymes and other proteins needed for growth. Then the cell moves to the S phase, which stands for synthesis. A signal has been given for the cell to begin dividing, so it has to replicate. That's synthesizing its DNA. It's a shorter phase. And then G2, the DNA has been replicated, so now it needs to be checked. Repairs are made. The cell continues to grow and replicate the organelles. It's going to be prepared to divide. There is a sharp increase in the protein tubulin, which is needed to make microtubules for the spindle fibers that are needed during mitosis. After interphase, the cell enters into the M or mitosis phase. Growth temporarily stops, and then the nucleus divides into two daughter nuclei. There are four main parts to mitosis: prophase, metaphase, anaphase, and telophase. Following mitosis, the cell then goes into cytokinesis, where the whole cell divides. In an animal cell, the division involves the constriction of the cytoplasm between the two nuclei. In plant cells, it involves the formation of the cell wall. The length of time that a cell spends in the cell cycles varies, depends on the environmental conditions and the type of cell. Root tip cells of onions, they divide about every 20 hours. Epithelial cells found in the human intestines, they divide about every 10 hours. Mitosis is a continuous process. It's divided into stages for convenience. We use PMAT to help us remember just the little acronym: prophase, metaphase, anaphase, and telophase. In early prophase, the chromosomes start to coil up and become visible when stained. Late prophase, centrioles move to opposite sides of the nucleus. The nuclear envelope breaks up into vesicles, which makes it invisible at that time. A lot of times you'll see where it says the nuclear envelope disappears. It's because it's in a vesicle. We can no longer see it. And then the chromosomes are seen as identical chromatids. Metaphase, the centrioles reach opposite ends or opposite poles and organize spindle microtubules. Chromosomes align at the equator of the spindle, so the microtubules can attach to the centromeres. Anaphase, the chromatids move to opposite poles. They're pulled by the centromeres. And then in telophase, the chromatids have reached the spindle and begin to uncoil. The nuclear envelope reforms. We will look at the stages of mitosis in more detail during lab. The cell cycle concludes with cytokinesis, which is the dividing of the cytoplasm. Again, in animal cells, the cell divides by the constriction of the cytoplasm. As the cell changes shape, the surface area of the cell increases as it's going to form two new cells. In plant cells, they do not contain the centrosomes. After the nuclear division of a plant cell, a new cell wall is going to form between the two daughter nuclei. This is just given a rundown of the stages of mitosis, going through prophase, metaphase, anaphase, and telophase in an animal cell. Okay, centrosomes, centrioles, and centromeres. 3 vocab words that we need to make sure that we keep straight. Centrosomes are located at the poles of the spindles, one at each pole. They're found in animal cells, and they act as microtubule organizing centers. Each centrosome consists of a pair of centrioles surrounded by a large number of proteins, which controls the production of microtubules. The centromere holds the chromatids together and is involved in the separation of the chromatids during mitosis. In mitosis, the centromere is the site of attachment of the spindle microtubules. The centromere holds chromatids together and is involved in the separation of those chromatids during mitosis. In mitosis, the centromere is the site of attachment of the spindle microtubules. So you have your centromere. It's holding the sister chromatids together. And then the microtubules are attached so that as these condense and shorten, it's going to pull those sister chromatids apart. Each metaphase chromosome has 2 kinetochores at the centromere, 1 on each chromatid, which you can see in the diagram. The kinetochores are made of protein molecules which connect centromere to the spindle microtubules. Kinetochores develop before nuclear division starts and are lost afterwards. Another question for you guys to answer. This one might require you having to go back and look at chapter 1. Looking at some of the importances of mitosis. Growth. Daughter cells have the same number of chromosomes as the parent cell and are genetic clones. This allows growth of multicellular organisms from a unicellular zygote. Growth can occur within the entire body, such as in animals, or confined to a certain region, as in the meristems of plants, meaning like the root tips growing in the onion. Replacement of cells and repair of tissues. Cells are constantly dying and have to be replaced by new cells, such as replacing your skin or the lining of the gut. Some animals can regenerate entire body parts. A starfish can regenerate new arms. Asexual reproduction is the production of new individuals of a species by a single parent organism. The offspring are genetically identical to the parent. This can be from a unicellular organism such as an amoeba or multicellular, as in budding of a plant. Budding of a plant is when you take a part of the plant, forms like a little nub, and it comes off, and then you can put it into the soil and it grows into a whole new plant. The ability to generate whole organisms from single cells or small groups of cells is important in biotechnology and genetic engineering and the basis for cloning. Cloning of the B lymphocytes and the T lymphocytes during the immune response is dependent on mitosis. We will have more of this in a later chapter. Some questions for you guys to answer. If you would pause the video and work on these questions before proceeding through the notes. The role of the telomeres. Remember, the telomeres were at the ends of the chromosomes. They have an important function. You can kind of think of telomeres kind of like the plastic tips on shoelaces. On the ends of the shoelaces, how you have those little plastic pieces that hold the shoelace together. Shoelace is good until those plastic pieces are gone, and once they're gone, the shoelace falls apart. Well, the telomeres are going to ensure that the ends of the molecule are included in the replication and not left out when DNA is replicated. Telomeres work by making the DNA a bit longer. They allow the copying enzyme to complete the copying for all of the meaningful DNA. The enzyme that's responsible for this is the telomerase. Telomeres' function is to prevent the loss of genes during cell division and allow continued replication of the cell. A lot of research going into looking at the role of the telomeres and ways to make sure that the telomeres do not shorten as cells divide. Stem cells are also important in research. A stem cell is a cell that can divide an unlimited number of times. Each time it divides, the new cell has the potential to remain a stem cell or to develop, meaning differentiate, into a specialized cell, such as a blood cell or a muscle cell. The power of a stem cell to produce different types of cells are variable and referred to as its potency. Stem cells that produce any type of cell are described as topoisotent. The zygote is topoisotent, as are all the cells up to the 16th cell stage of development. At that point, some cells produce the placenta, while others are going to develop the embryo. There are more than 200 different types of cells in an adult human body. All right, another question for you to answer before finishing your notes. In adults, the more specialized a cell, the more they lose the ability to divide. However, it's important for growth and repair, so a small population of stem cells remains. Stem cells found in bone marrow are multipotent. They can produce red blood cells, monocytes, neutrophils, and lymphocytes. Blood cells have a short lifespan, around 250 billion red blood cells and 20 billion white. Blood cells are lost and need to be replaced daily. In adults, stem cells are found in bone marrow, skin, gut, heart, and brain. This opens the door for future medical practices, studying things for blood cancers such as leukemia. There's a lot of hope for diabetes, muscles and nerve damage, and brain disorders such as Parkinson's and Huntington's disease.